My name is Januário, and this is my scientific initiation project, Isolation and Study of Growth Promotion by Dark Septate Fungi from Yerba Mate. First, I want to highlight the importance of yerba mate. It is widely cultivated in Brazil. Brazilian production in 2023 was of about 425.8 thousand tons, and the production value was of about 589.6 million reais. However, its production still faces some challenges, such as susceptibility to biotic and abiotic stresses, and endophytic fungi, particularly dark septate endophytes or DSE, have gained attention due to their ability to establish beneficial interactions with plant roots, promoting tolerance to adverse environmental conditions. My study aimed to isolate and characterize fungal strains obtained from yerba mate roots, assess whether they present characteristics compatible with those of DSE, and investigate their potential in promoting growth and stress tolerance. I collected the roots, as can be seen in this image. The roots were disinfested, sectioned, and plated on culture medium. I isolated the fungi and then subjected them to light culture techniques to allow observation of fungal structures using light microscopy. Additionally, identification was performed using Malditoff mass spectrometry. The results included microscopic images of the isolates, which enabled the analysis of spore, hyphal, and conidiophore morphology, along with identification data obtained via Malditoff. The results, so I brought here one example of one of the isolates. In image A, we have the light culture, and in image B and C, we have the light microscopy of one of the isolates. And for this particular isolate, the result of the Malditoff was that it was of the species Aspergillus niger. I was able to observe that there were some discrepancies between what I studied through the morphology of the isolates and what the Malditoff got us as a result. And we know that the Malditoff is fast and low cost, but it has its limitations. We saw that the results of the Malditoff were not compatible with what we were observing in the morphology of the isolates through light microscopy. So here is one example. In the left is the microscopy I got in this experiment, and in the right an image of the Fusarium solani gotten from a scientific article, and we can see there are a lot of similarities. So the spores are banana shaped, and there are these points of accumulation of spores that are very characteristic. And here we got in the right an image of the Aspergillus niger, which is the species that the Malditoff brought as being what of the isolate we got, that is here in the image in the left. And we can see that there are a lot of discrepancies. They are not compatible, they have not the same characteristics. So I was able to conclude that the Malditoff is useful, but not enough by itself. So I highlight the importance of the optical microscopy and morphological analysis, which remain essential. I saw that the Malditoff generated inaccurate results, leading to the misidentification of the isolated fungi. And none of the isolates of this project were characterized as DSE. So for future perspectives, I could try to isolate the fungi once again to try to find an isolate compatible with the characteristics of DSE.